Hello, and welcome to this series on DNA variant interpretation. I'm Sam Strome, PhD, Assistant Professor of Pathology and Laboratory Medicine at UCLA. Genetics and genomics are already becoming a part of everyday life in the world of medicine. Because the technology to perform genomic testing is so new, most medical professionals, even geneticists, have little or no training in the area of DNA variant interpretation. Unfortunately, there are very few opportunities to gain such training. This video is aimed at students, doctors, nurses, genetic counselors, and other medical and research professionals interested in increasing their knowledge about the relationship between genetics and health. You may soon find yourself faced with a patient who has a complex genetic test result and needs help interpreting what those results mean for their health and for the health of their family. Or you may one day be such a patient. In either case, I hope this series helps put genetic test results into context and gives you some tools to understand them better. A few ground rules before we get started. First off, you should understand the central dogma of biology, that DNA is an information storage molecule for genes, which is copied into intermediate messages called RNA, which in turn are processed into proteins, which do the work of the cell. There are many exceptions to this framework, but it's a very useful jumping-off point for learning genetics and genomics. Second, natural selection is the mechanism of evolution, which has given rise to all forms of life on Earth and continues to shape it. If either of these ideas is foreign to you, there are plenty of resources available to learn more. I particularly recommend the book The Beak of the Finch by Jonathan Weiner. It's an exceptionally well-written documentation of scientists who studied the birds of the Galapagos Islands and produced some of the world's most powerful evidence of evolution by natural selection. I also recommend a personal favorite, Carl Sagan's book and television series, Cosmos, if you want to hear passionate and eloquent explanations of these concepts and many others. Seriously, both of these are pretty awesome. Our DNA, the human genome, consists of about 6 billion DNA nucleotides, spread across 23 pairs of chromosomes, shown here in different colors. We all get one of each type of chromosome from our father, and one from our mother. One of these pairs is always the sex chromosomes, 2 copies of chromosome X for females, and 1 each of chromosomes X and Y for males. The remaining 22 pairs of chromosomes are called autosomes. As the result of a monumental worldwide effort called the Human Genome Project, we have had a rough draft of the human genome sequence since 2001. This rough draft has been revised and updated several times as more and better data have become available. The result is an incredibly rich resource, a reference sequence of a typical human genome, which we call HG19. A DNA variant is any place in a person's genome that differs from this reference. The concept of alleles is crucial to understanding genetics, and I want to take a moment to make sure the idea is clear before we go on any further. A single chromosome is made up of 2 DNA molecules bound together in the classic double helix. One strand is a perfect mirror image of the other. But remember that chromosomes come in pairs. Each cell has 2 different copies of each chromosome. That's 4 total DNA molecules for each chromosome. Unless you're a fancy dog breed, these 2 chromosomes are not identical. They are called sister chromosomes, and one came from mom and one came from dad. So for each autosome, the non-sex chromosomes, there are 4 molecules of DNA arranged in 2 sets that are a mirror image of each other. An allele is like a flavor of a gene, defined by the variants in it, and each person has 2 alleles of every gene. If both alleles match the reference sequence, that's called homozygous reference, or wild type. If a variant is present on both alleles, that's called homozygous. If a variant is present on one allele but not the other, that's called heterozygous. If there are 2 different alleles present on opposite chromosomes but in the same gene, the 2 variants together are referred to as compound heterozygous. DNA of humans and our closest living relative, the chimpanzee, is about 98% identical. Between any 2 humans, there are even fewer differences. Less than 1% of the DNA nucleotides differ between your genome and mine, or your pen pal halfway across the world. What you're looking at right now is an installation designed by artist Michael Marcovici, which contains 1 billion, with a B, dollars in cash. That is a lot of money. If you were to, say, borrow 1% of that $1 billion, that would be $1 million, which is still a lot of money. Similarly, because the human genome is so large, that small fraction of variation adds up. Typical human genome has between 4 to 10 million variants compared with the reference. That's about 1 variant per 1,000 nucleotides on average. There are many kinds of DNA variants, ranging from the very large to the very small. The first DNA variants discovered were the aneuploidies. This is when an entire chromosome is missing or has too many copies. An example of this is Down syndrome, which results from having 3 copies of chromosome 21. Such variants can be detected by karyotype, which is essentially looking at chromosomes under a microscope. Most people do not have any DNA variants of this type in their genome. Other types of DNA variants include large deletions or duplications of pieces of chromosomes. These are called submicroscopic deletions or duplications, as in they can't be seen in the microscope. A test called chromosomal microarray analysis can be performed to look for these changes, which result in the gain or loss of many genes at a time. The average person has maybe 1 or 2 such variants, if any. The smallest DNA variants include single nucleotide variants and small insertion deletions, or indels, which are typically between 1 and 20 extra or missing nucleotides. An example of a small deletion is shown as the dashed line in the bottom right image. The bulk of DNA variants are either single nucleotide changes or small indels. The best tool for identifying new single nucleotide variants or indels is DNA sequencing. This table shows the major types of DNA variation and some basic information about each. I won't go into much detail right now, so you may want to do a screen grab and save it for later. If you don't know how to do a screen grab, on PC just hit the print screen button to copy the screen image. On Mac, click the magnifying glass in the upper right-hand corner, type in the word grab, hit enter, and then hit Command-Z to copy the image to your clipboard. On either platform, once you've got the image copied, paste it into a program like Microsoft Word or PowerPoint. There. Now you can't say I didn't teach you something. And back to genetics. Imagine typing out a 6 billion letter book, and doing that a few million times. You'd probably end up with more than a few typos. Analogous challenges face dividing cells, including sperm and egg. Incredible DNA proofreading mechanisms have evolved, which minimize the volume of copying errors, but these are imperfect. In the text-based analogy shown here, the spell checker has identified that the first word has been misspelled, but didn't notice the last word, which is probably also wrong. Some natural and unavoidable things like UV light and cosmic radiation, as well as some chemical exposures, can cause DNA damage resulting in changes. The proper name for a new DNA change is mutation. All told, the average person probably has around 100 or so mutations compared to their parents. You've probably heard that all the cells in your body have the same DNA. For the most part, that's true. The mutations I'm talking about here are the ones that actually occurred in the sperm or egg cells of your parent. So they ended up in all the cells of your fetal self and will persist throughout your entire life. But all DNA everywhere is constantly mutating at a slow rate, especially in dividing cells such as your skin or your intestinal lining. When those kinds of mutations build up and cause cells to grow out of control, we call it cancer. I'll have much more to say about that in later lectures in this series. So if there are only about 100 new mutations in every person, what about those millions of other variants that I described earlier that occur in every genome? These variants are inherited from previous generations. They were once new mutations in a distant ancestor, which have been passed on down their line, from their children to their grandchildren and beyond. Let's consider a group of 10 people, each represented by a box. One of them, shown in red, has a new mutation. People go on to parent a total of 10 children. Every time the red guy mates, he has a 50% chance of passing on the new mutation because he possesses one normal chromosome and one chromosome with mutation on. In this example, he happened to pass on the red mutation to both of his kids, which is not at all unlikely. Now, purely by chance, 20% of the population has the red mutation. Actually, in this second generation, this thing isn't a mutation anymore. Because it's observed as inherited by multiple individuals, it is now simply a DNA variant, just like the other millions of variants across the genome. We can carry this forward a few more generations to the great-great-grandchildren of the original red box. By following the red lines, you can tell that 6 of the people in the youngest generation are direct descendants of the original red guy, and 3 of them have that red variant. This type of thing is happening all the time, and with 7 billion human beings out there, there are plenty of opportunities for variants to get passed around. So DNA variants are born as mutations, and live on in future generations as DNA variants. Some DNA variants are improvements over previous genomes. For example, a DNA variant might result in a digit to be separated further from the others, which could result in an opposable thumb. Other DNA variants may do nothing at all. Unlike bacteria and other small organisms which have supremely efficient genomes, big creatures like primates have large swaths of DNA which appear to be unfazed by small changes like single nucleotide variants or small indels. Some variants may cause changes like different eye color or hair color, which increase the variation of a species but don't necessarily have an obvious effect on genetic fitness. Other variants make things worse. These are the types of variants that are most interesting for medical purposes. The goal of DNA variant interpretation is to tease out which variants fit into that last category, and how they may affect the current or future health of an individual and their family. Thank you so much for watching. The next installment of this series will focus on interpretation of DNA variants for individuals with rare genetic conditions using gene panels and exome sequencing. If you have any questions or comments, please contact me on Twitter at StromDNA. As Carl Sagan said, these are some of the things hydrogen atoms do, given 15 billion years of cosmic evolution. Thanks, and have a great day.